Hello, everyone. Thank you for joining me today. My name's Daniel Wu, and I'm an AI executive educator and author. My most recent role was with JP Morgan as their head of AI machine learning for commercial banking. Um, and I'm looking for my next mountain to climb. Um, I'm a course facilitator for Stanford's AI professional program, and I also have the pleasure of co-authoring two recent AI books. Last year was on Gen AI security, and this year is about Agentic AI, as you can see here. Later, I'll share the information. Some of the content is discussed in the book as well. So I'm thrilled to be here to discuss a topic that I'm incredibly passionate about, and if which is accelerating scientific discovery with agentic AI. And before we start, if you could please take a few seconds to kind of respond to the poll that's available. I'll leave that open for a little bit, maybe two more minutes, and then we can look at the results. Um, that would be great. Thank you. So, scientific discovery is one of humanity's greatest endeavors. It's often very slow, expensive, and it's also limited by human bandwidth. So, today we're going to explore how a new paradigm in AI is positioned to revolutionize the scientific landscape, tackling some of the most complex and costly challenges we face. So to set the stage, that's considered a challenge that perfectly illustrates the scale of the problem we're up against, the drug development process. Um, I don't know how many of you are familiar with this, but as you can see from this infographic, bringing a new drug to market is a monumental undertaking, to say the least. We first start with a massive pool of 10 to 15,000 compounds. And over a period of 3 to 6 years of drug discovery and preclinical trials, that number is whittled down to just 250 compounds deemed promising enough for further investigation. And then from there, we enter the lengthy and expensive phase of clinical trials, which can take 6 to 7 years. Often, five of these 250 compounds are considered safe enough for testing in human volunteers. And of those five, typically only one will ultimately receive FDA approval and make it to the market. So as you can see, the entire process from initial discovery to an approved drug takes an average of 10 to 15 years and costs over $1 billion, sometimes approaching $2.6 billion if you account for failures along these process. So this is the kind of high stake, long horizon problem where we desperately need a breakthrough. And this is where agentic AI comes in. So with that, let's take a look at the at our poll results. So thank you for responding to the result. So about 63% you're exploring about the potential AI and generative AI. This is very common. This is kind of aligned with what I expect to see. Yeah, there's a certain organization already pretty ahead of time actually start building agentic AI systems. So those are typically companies, organizations that has started their AI journey way before this year. But this year, the talk of the town is all about agentic AI. So great. So this means that there's a lot of potential that this talk may inspire you to start exploring this paradigm shift in agentic AI. Thank you for participating. Um, let's go back to our presentation. All right. So, what is agentic AI? Why is it so a hot topic this year? So, and how does it differ from the AI we're already familiar with, years back? So, for the most part, we have been interacting with AI as a predictive tool, which is shown on the left side. What that means is simply that we provide an input to the AI system and it generates an output for us. And think of a model that predicts a protein folding structure or identifies potential drug candidates from a list. Agentic AI platform, however, represents a very significant paradigm shift. Instead of just performing a specific predefined task, an agentic system can reason, plan, and execute a sequence of actions to achieve a complex goal. It can interact with its environment, use some tools, and even collaborate with humans to get the job done. So, unlike traditional AI, agentic AI systems are designed to operate with a high degree of autonomy. This allows them to independently perform tasks such as hypothesis generation, literature review, experimental design, and data analysis, all very critical phases in scientific discovery. And these systems have the potential to significantly accelerate scientific research, reducing cost and expanding the access to advanced tools across various fields, including chemistry, biology, and material science. So within the world of agentic AI, we can further distinguish between single agent and multi-agent systems. So a single agent system is able to achieve its goal independently without relying on assistance or feedback from other AI agents, even if there are multiple agents coexist within the same environment. However, there may be opportunities for humans to be in the loop as well by providing feedback for agent guidance. Now, in contrast, multi-agent systems, on the other hand, involve two or more agents interacting with each other, and they are particularly powerful in complex environments. And in this setup, each agent is an expert in a specific domain. And these specialized agents can then collaborate to solve multifaceted problems, which is very common in scientific discovery. However, managing the communication and interaction between all these different agents can be a significant technical challenge by nature. So, another key distinction is how these agents interact with us, the users, the humans. So, we can think of it as a spectrum from full autonomy to human AI collaboration. So, fully autonomous systems are designed to operate independently and they will automate end-to-end scientific workflows with minimum human intervention. And these systems leverage advanced AI capabilities such as natural language understanding, planning, decision making to perform very complex tasks ranging from hypothesis generation to experiment execution. And as you can see, the main advantage of these fully autonomous system is their efficiency in environments where tasks are well defined, repetitive, or require high precision. They can significantly accelerate research by automating the time consuming processes. Now, on the flip side, they may struggle with tasks that require creativity, domain specific intuition, or some complex, interdisciplinary knowledge. So this basically highlights the need for human oversight in certain scenarios. And this is where human AI collaboration, or what I call augmentation, becomes very critical. Human AI collaborative system emphasize the synergy between AI and researchers. And this type of systems will combine the computational power of AI with the creativity and human expertise. Advantages of these AI-driven augmentation lie in their ability to accelerate research, enhance experimental design, and optimize decision making in fields like genetics, material science, and chemistry. Now, they also have limitation. The limitations stem from their reliance on human oversight, and also their performance highly depend on the underlying data quality and interpretability. So they still require manual validation and execution. So with that, let's look at what this looks like in practice. So this diagram shows a generalized workflow for agentic AI in scientific discovery. It is a continuous loop of thought, action, observation, and this allows the AI to interact with tools and physical world to carry out complex scientific tasks. So ideation phase. Ideation basically is the process of generating, refining, selecting research ideas or hypotheses. And AI agents can automate this process by analyzing existing literature, identifying gaps, and proposing novel hypotheses. And hence, they can accelerate the initial stage of research. Now, during experimentation, design, and execution, AI agents autonomously can design and execute complex experiments by integrating tools for planning, optimization, and robotic automation. Now, during data analysis and interpretation, AI agents can process large data sets and generate insights that might be overlooked by researchers. And this enhances the accuracy and efficiency of this stage. And finally, during the paper writing and knowledge dissemination phase, AI agents basically automate the writing of research papers, make sure there's clarity, coherence, and adherence to academic standards, also reduce the time and effort required for publication. So with this in mind, let's dive into two very specific examples or use cases to highlight how AI agents actually accelerate scientific discovery. So our first use case is a system called CameCrow, and this was a paper published, and this is published in Nature last year. Um, and Kim Crow is an AI agent in chemistry. So it gets its name, fun fact, it gets its name by combining chemistry with crow. And crow, as most people know, that symbolize very intelligent bird that knows how to use tools to get the task done. So standard LLMs are designed to predict the next token in a sequence. And that's not necessary to be factually accurate in highly technical domains such as science field. And this means that they can't reliably do things like convert a chemical's name to its structure or predict a reaction outcomes. So this fundamental limitation makes them untrustworthy for serious scientific applications on their own. So Kim Crow's approach is not to build a new LM from scratch, but to augment an existing one. So by connecting GPT-4 to specialized software tools like a calculator for math or a specific chemistry program for molecular analysis, CameCrow can ground the LM's reasoning in factual accurate data. This creates a very powerful synergy. The OM will act as a reasoning aid engine that understands the user's goal, while the tools will provide the necessary domain-specific knowledge and capabilities. This allows CameCrow to tackle diverse tasks across organic synthesis, drug discovery, and material design. So if I take a closer look at how this works, so CameCrow's power lies in its think and act methodology. At its core is GPT-4, which provides the reasoning and planning capabilities. So this operates within a thought, action, observation loop. The methodology is basically inspired by a framework like REACT, stands for reasoning and action, which combines chain of thought reasoning with tool use. And the user will provide the task, like synthesizing an insect repellent. CamCrow then thinks, reasons, and plans a sequence of actions. It will select and use tools from its specialized toolbox, which includes tools for molecules, reactions, safety, and general research tasks. It then observes the results and continues the loop until the task is complete, even in enabling autonomous experimentation. So if you're interested, I would encourage you to look into this Nature paper to read more. And they have very kind of fascinating example about asking Cane Crow to synthesize insect repellent, and Cane Crow basically successfully synthesized and in the lab synthesize the DEET molecule. So this example is very inspiring in a sense and encouraging and applying agentic AI in this field. So let's take a look at the result reported in the paper. The results are impressive. Ken Crow has been proven very effective at autonomously synthesize the insect repellent DEET and three different organocatalysts. It has also demonstrated the power of human AI collaboration by helping researchers discover a novel chromophore. So the successful synthesis of these four distinct molecules demonstrates that CameCrow is not just a theoretical concept. It can actually manage a full workflow from a simple text prompt to a successfully executed chemical reaction without human intervention. The evaluation result compared CameCrow and standard GPT-4 across 14 different tasks. While GPT-4 performed adequately on simple memorization tasks, it failed on novel or complex ones. CameCrow, using its tools, consistently provided better solutions for these more challenging and useful use cases. So an interesting finding was also the failure of LM-based evaluation. They use an evaluator LM called Evaluator GPT, which actually preferred the confident but incorrect responses generated by GPT-4 over CameCrow's more cautious tool-verified answers. So what this shows is that for scientific fact finding, human expert evaluation still is the gold standard for now. And I know it's very popular out there to apply automatic evaluation using LMs, and but at these high stake use cases, really nothing beats the expert evaluation as the gold standard. So quickly summarize, CameCrow's strengths are its ability to reduce hallucination, automate complex workflows, and incorporate built-in safety checks. However, it's not without its challenges. The system is highly dependent on the quality and breadth of its tools. And like all current models, since it relies on LLMs for reasoning, it has that limitation of the reasoning capability is limited by the underlying LLM's reasoning capability. And future direction for CamCrow include expanding its toolbox and improving reasoning with more advanced LLMs, and also developing better standardized benchmarks for evaluation. Now let's turn our attention to another complex scientific domain, gene editing. And this again is another paper published this year in Nature. CRISPR is a gene editing technology adapted from a bacterial immune system. It allows scientists to precisely target and modify DNA sequences in living cells and organisms. And this technology has revolutionized biomedical research and offers potential for treating genetic diseases, improving agriculture, and more. So, designing effective CRISPR experiments requires deep expertise in both the technology itself and the specific biological system being studied. This is another area where general purpose LLMs often fall short. CRISPR-GPT is an AI co-pilot that integrates domain expertise from published literature, retrieval techniques for accessing information, and external bioinformatic tools. And this allows it to function as a knowledgeable partner, guiding researchers through complex workflows and handling technical tasks, and ultimately resulted in validated real-world experiments. So let's take a look at how this works. So CRISPR-GPT use a different approach. It utilizes a sophisticated multi-agent architecture. So you will have LM planner agent, which basically decomposes a user's high-level request, such as knock out gene X, into a logical sequence of discrete tasks. Then we have task executor agent, that executes each task using state machines, which are structured workflows that guide the process step by step and call external tools when needed. And then you have tool provider agents, which provides access to essential external tools, including web search like Google, literature search like Google Scholar, and bioinformatics software. Finally, we have LM user proxy agents. These agents act as the interface, interacting with the user for input and communicating the system's actions and results, which will enable human oversight and collaboration. So this architecture also allows for a high degree of flexibility, offering different levels of automation to cater to users with varying experience. Um, and expertise. So you have the meta mode that provides step-by-step guidance through predefined end-to-end experimental workflows. And this is an ideal mode for beginners. You also have auto mode, which dynamically builds and executes a custom workflow based on a freestyle user request. And this is suited for advanced researchers. Finally, you have the Q&A mode, which provides real-time on-demand answers to ad hoc scientific questions about gene editing. So if you look at this very sophisticated multi-agent state machine-driven approach, makes it makes the system very robust and transparent. The users can also monitor the process, providing corrections and understand the reasoning behind each decision. Eventually, it fosters a true human AI collaborative environment. So the results reported in the paper are equally, if not more striking, compared to the Cane Crow paper. It has been shown to be incredibly effective, guiding junior researchers without any prior gene editing experience to successfully complete their complex experiments on their very first attempt. So that is impressive. The standardized gene editing benchmark, which contains a set of 288 test cases, CRISPR-GPT achieved accuracy, precision, recall, and F1 scores of over 99% or 0.99. Now, in a blinded study, there was a panel of 8 gene editing experts also rated CRISPR-GPT as more accurate, more complete, and concise than baseline LLMs. Experts noted that CRISPR-GPT provided detailed guidance with no hallucinations, whereas general LLMs made factual errors and lacked the ability to perform actions without bioinformatic tools. So this demonstrates the immense value of a specialized agentic approach for complex scientific tasks such as gene editing. So in summary, CRISPR-GPT's core strength is its ability to deliver accurate, actionable guidance in a complex scientific domain, effectively acting as an AI co-pilot that enhances research quality and reproducibility. It also has built-in ethical guardrails as a critical feature. The system actively screens for requests that's related to editing heritable human genes or dangerous viruses and includes filters to protect patient privacy. This sets a standard for safety in scientific AI tools. However, the system also has some limitations. Its knowledge is ultimately derived from human experts, and the manual effort required to create and curate this knowledge base to drive this agentic platform makes it difficult to scale. As with any AI, its performance is only as good as the data it is trained on and the tools it has access to. Looking forward, we can see the vision for CRISPR-GPT extends beyond just computational design. The tool is also to bridge the gap between digital design and physical execution by connecting the agent to robotic lab systems, such as the one that's used in CameCrow. This would enable researchers to orchestrate experiments from their computers, with robots performing manual lab work. This will minimize human intervention and dramatically accelerate the cycle of scientific discovery. And in addition, the system's modular architecture allows for easy integration of new tools and functionalities beyond gene editing, such as uh, plasmid design or protein foundation models. So what does this mean? How do we generalize what we learned from these two papers or these two successful use cases? Some key strategic takeaways are as follows. Number one, the role of scientist and engineer is changing. The primary skill is shifting from doing the tedious work to asking the right questions. The value is formulating the hypothesis for the AI agent to investigate, and this applies to all technical fields. The second takeaway is I would say your data infrastructure essentially is your lab. What this means is the success of these agents is entirely dependent on the access to high quality, well-structured data and tools. For tech leaders, this means investing in robust data platforms and API first architectures. It is no longer optional, it's foundational to leveraging agentic AI. And finally, governance and validation are non-negotiable. These AI agents are very powerful, but they are not infallible. In high stakes fields like science and finance, security, healthcare, you name it, you must have a human in the loop for validation. The tech leader's role is to design such workflows that combine AI speed with human expertise and ethical oversight. So, with that, we're just about at time. I'll just to sum up, the era of agentic AI is here, and applying agentic AI in science is just beginning. But it holds the promise of dramatically accelerating the pace of discovery and helping us solve some of humanity's most pressing challenges. Thank you for your time and attention. On the left side of the screen is the QR code for my LinkedIn profile. If you'd like to, I'll be happy to connect after the event. On the right of the screen is the QR code for the Amazon page of the recently published Agentic AI book that I had the pleasure of co-authoring. It was published back in June. If you'd be interested in exploring more, we talk more about agentic use case across different industries and provided some strategic framework for decision makers to consider as well. So again, thank you for your time and attention. With that, I think we're running out of time, um, but I'm happy to follow up with any questions you may have. Thank you.